Recording pause mode is impossible.
Yeah. Yeah, Roopa, start sharing your screen. Go to the first slide.
Is it visible, sir?
Yeah, it's visible now.
Okay.
So hi everyone. It's good to see like there are 41 students who have joined this class. So today we are going to start the plan reading for ICAR JRF Plan Sciences. So we have a new mentor or educator here. She is my junior, Roopa. She will be introducing herself now and she will be continuing the class. So since this is our first class, so be interactive, ask your doubts, and wherever you feel uncomfortable or if you have any queries, you can stop her and clarify all your queries. Okay. So since the plan reading is very much important for you and in the exams, it carries a good weightage. So be attentive and concentrate on the class and clear all your doubts even before or after the class, and we'll be also providing the notes and the mock test after every class. So be in touch with the mentor so that all your doubts will be cleared. And advance happy new year to all of you. I hope like this year brings lots of joy, wealth and prosperity in your life, and I wish you all of you to clear your ICAR JRF exams for which you people are working hard.
Thank you, sir. We'll continue with the class.
Let's start.
Good evening, students. Myself, Roopa MN. So today will be our first class of plant breeding. So I welcome all of you to our online master course, Plant Sciences 2024. So before starting, from today, we will be starting your plant breeding classes. Before starting this class, let's have a brief introduction about myself. So I'm your educator, Roopa MN. So I have done my undergraduation from College of Agriculture, Mandya, University of Agricultural Sciences, Bangalore. So I have done my master's in genetics and plant breeding from Indian Agricultural Research Institute, New Delhi. And during my education period, my achievements were I was awarded with 8 gold medals during my UG, and also I have secured All India rank of 6 during my JRF examination. Recently, I have cleared ASRB net also, and in my SRF examination, I have secured 28th rank. So currently, I'm not enrolled in PhD course. So I'm taking my examination in next year also. So meanwhile, I'll be helping you guys to prepare for your exam also along with me. So this is about my introduction. So let's get started. So plant breeding. How many of you have read this? Have started this plant breeding? So raise your hands. Okay. So some of you have started reading already. So this class is like a quick question and answer session for you guys then. Okay. Okay, let's get started. We'll see how much you have prepared. So what do you mean by plant breeding? Anybody can define it? Come on, I need answers. Can anybody define plant breeding?
Yes, ma'am. It is a science and technique in which we increase the quantity and quality of the plants.
Quantity and quality of the plants. Okay. Some more precise.
Ma'am, now it's not coming in my mind. Sorry.
Okay. Anybody else?
Manipulation in plants to obtain new varieties with desired characteristics.
Yeah. Okay. Good.
We try to change the characters of plants.
Yeah. Yeah, it's good that you guys are trying to answer. But it is like it is a set of methods and the principles we apply to the plant to change its genetic character in order to change it in terms of our favorable nature. Like we want some high yield or we want some disease resistance in that plants. So we change that plant according to our favorable nature. So for that, we will use certain principles and the methods. Those set of principles and methods, we call it as a plant breeding. Okay. Anybody knows any other definition for this plant breeding, which is defined by Vavilo?
Plant evolution directed by man.
Yeah, very good. It is a plant evolution directed by man. Okay. As you can see here, plant breeding is nothing but it is a principles and methods which are required for favorably changing the genetic constitution of the crop plants, or it is also a plant evolution directed by man. This statement is given by Vavilo. Make a note of it. Okay. So as we look into the history, the food grain production has increased from 50 million tons to 315 million tons as far as 2021-2022. Oh, can anybody guess what might be the reason for this drastic increase? There is approximately a 6-fold of increase in the yield. What might be the reason for this?
Green Revolution.
Green Revolution. What they did in the Green Revolution? Can you be
Yielding
more precise?
varieties were
Yielding varieties. Okay. What else?
Dwarf varieties.
What? Dwarf varieties. Okay. Dwarf varieties. Okay. What else?
Improved variety for disease resistant.
Yeah, they are all comes in a single class. Okay. Improved varieties. Then one more thing is there. You guys are forgetting it. Along with this improved varieties, they did something. Okay. It is a better management of the inputs. It is a better management of the inputs. Whatever fertilizers or seeds, whatever was there, they used it, whatever the cultural practices or plant protection chemicals, they used it optimizedly. And also along with that, they have changed the genotype of the plant in a way that it will yield us more. These are the two main reasons we can, when we look back to the history, we can make a note of it, that there is an improved use of the fertilizers and whatever the inputs along with the change of the genotype of the plants. So as you can see here, it is a better management of the inputs like fertilizers, irrigation water, plant protection chemicals, and also continuously favorably we are changing the genotype of the crop plants. Then if we want to look at the history of the plant breeding, so we will divide this, we will see the history first. Okay, so we will divide this history into different phases. So does anybody know what are the phases? There are 5 phases.
Domestication,
Yeah.
selection,
Yeah. Domestication.
hybridization,
Hybridization.
utilization of genetic variation.
Okay, genetic variations.
Mm,
And
Germplasm.
huh?
Genetic engineering.
Yeah, genetic engineering. Very good. Okay. So let us see one by one what is these phases are. What is domestication?
Bringing wild species under human management.
Yeah, very good. It is bringing the wild species under human management. Okay. Domestication is considered as one of the most basic method of the plant breeding. Okay. Why do you think so? Why it is the most basic method? Make a note of this point. This might be asked in your exam, which is the most basic method of the plant breeding. Okay. So why do you think it is the most basic thing?
Ma'am, because of domestication, the plant species are easily available for manipulation or for plant breeding.
Yeah, very good. So whatever the wild species which are there, human has brought it into the his cropland, we can say. After he has brought some crops into his cropland, only we could able to make some changes in those available croplands. We can say that. That's why it is considered as most basic method. Even today, this method is still available. We can see that. If we don't have any favorable varieties with us, we will go for some exploration phase and we will bring some wild species. We will domesticate it and we will make some changes in those genotypes. That's why it is considered as a most basic method. Whatever we do, it is after the domestication only. Okay. Next phase is the selection phase. What is selection?
Choosing of plants with
Ma'am, if
desirable
we consider
characteristics.
a population, there will be a difference in reproduction rate of fittest type, the
Yeah.
fittest and the less fittest type. The
Yeah.
reproductive rate of fittest will be more, the progeny will be more as compared to the inferior one.
Yeah, very good. It is a differential rate of reproduction of different genotypes. Okay. So we will select the genotype which is more superior and we will allow it to reproduce than the others. So we are creating some kind of differentiation in their reproduction ability so that those selected varieties will become more successful and will remain further. Okay, this is the selection. So from where this selection has started? Who is the first person who started selection? Does anybody know? When we look back to the history, it is from Patrick JRF. He has practiced some selection and he has given us two kind of statements. One thing, he has, even in the early phase itself, he has noticed that only the heritable variation will respond to the selection. Heritable variation will respond to selection. What do you mean by this? Only heritable variations will respond. Why not other?
Ma'am, because that will be inherited from one generation to another.
Yeah, for whatever the trait you select, say for tall plant, you select it for this trait. And if you want to see this trait even in your progeny also, when it will be possible? Only when that tall trait is heritable. Okay, then only we could see the trait in the next generations. So that particular thing is observed. And also, how does these variations has come? He has also noted on this point that how does these variations has come? That is only through the mutations. Okay, through the mutations and natural hybridizations. Okay, these mutations have come from the these changes have come from the mutation in the natural hybridization. He has reported this. So after him, another one more famous scientist comes, that is a Vilmorin. What is the contribution of Vilmorin? He is at 1856. Anybody know?
Progeny test.
Progeny test. Okay. Why do we need this progeny test?
Evaluation of parents.
Okay, evaluation of parents. What else? What does progeny test tell us? Anybody know?
Performance of the parent. Performance of the parents plant.
Sorry, I didn't hear you.
Performance of parent plant.
Okay, okay. You're almost correct. Like it is estimating the worth of the parent based on the performance of its progeny. So we will first see the progeny. If it is performing as its parent, then only we will say that the parent is worthy. Okay, it is estimating the worth of the parent. Based on performance of its progeny. On which crop he has worked, Vilmorin?
Sugar beet.
Sugar beet.
Yeah, sugar beet. He was trying to improve the sugar content of the sugar beet. Okay. Then he has observed that he has also worked with another crop. What was that? He was also working with wheat. What kind of crop it is, wheat? What kind of pollination it has? Self or cross?
Self-pollination.
Okay, it is a self-pollinated crop. It is a highly self-pollinated crop. So he was also working with the wheat as well as the sugar beet. What he has observed that in sugar beet, he has gone to about some more generations of about 30. Till then, he did not observe any of the variations, like any of the heritable variations. But in this, for a very little time, he has got the heritable variations in them. So based on that only, he has given this progeny test that in a self-pollinated crop, whatever the variations you see, and based on that variations, if you go for some selection, then that selection will not be helpful. It will not be helpful, okay? Because whatever the variations which arise in the self-pollinated crops, they doesn't have any genetic basis. It is only environmental. Why so? Because in the next generations, those variations will... Come again. Hello. Because those variations we could not able to see in the next generations. Only the heritable variations we could able to see. After this, one more scientist will come. And also through this progeny test, what we can see. Anybody can say what we can see from this progeny test? What is the use of this progeny test?
Ma'am, we can determine the value of genotype of plants based on the performance of progeny.
Okay, that is itself in the definition.
We can get to know the vigor of the progeny.
Vigor of the progeny. Okay. What else? We can able to see the breeding behavior of the plant, whether it is a breeding behavior, whether it is a self-pollinated crop or a cross-pollinated crop, we could able to see that. As I said, in wheat and this sugar beet, you could able to see the difference. Why? Because it is because of its breeding behavior. Okay. And also whether the selected plant trait, whatever the trait you have selected, right, that is heritable or not, you can say by using this progeny test. So after that, in this selection phase, one more scientist who comes is Nielsen Ely. So he has practiced, initially he has practiced the individual plant selection. He's one among them who has practiced this individual plant selection. What is his another contribution? One more famous contribution is there.
Multiple
factor hypothesis.
Yeah. Multiple factor hypothesis. You guys have read already everything. Hypothesis. This is very good. So one more person who comes with, along with Wilmorin only, you will read him. So Johansen. So he has given this pure line theory. What is pure line theory? What he did? What he has done in pure line theory?
Ma'am, he concluded through this theory that variation within pure lines or self-pollinated plants is not heritable. It's of no use.
Yeah, correct.
Is due to environment. And he used princess variety for his experiment of Rajma.
Yeah, very good. You made my job very easy. Yeah, he has given the genetic basis for this individual plant selection. Till then, they were saying that individual plant, when you select, there is no if there is no heritability in that, you won't get that variation. They were just making this statement. He has given this concrete evidence that why this individual plant selection will not be useful if it is not having any heritable variations. So that is given by Johansen. So he has worked in the princess variety of Rajma. So what he has done in this and all we will see in the further classes. It is like a brief introduction class that you will see the phases only. So you will get to see lot many scientists in your this breeding courses. So when and where that chapters comes, you will see all the scientists who are associated with that particular technique or whatever. So you learn in that chapter itself. Don't burden yourself trying to learn all the scientists at a single go. Okay, whatever you encounter in that chapters, you try to remember those. So next phase, hybridization phase. What is hybridization?
Process of creating genetic variation.
Okay, process of creating genetic variations. What we do in hybridization?
We take two inbreds and cross them, then we get F1, which is a hybrid.
Okay, not necessarily inbred. You can take whatever two varieties or whatever it is, you will cross them and you will get some variations. What to say, parent 1 and parent 2. So you will hybridize and get some F1. Okay, this is the hybridization. So what are all the scientists who will come in this hybridization? Means Babylonians and Azurians. You have heard about this? What their contribution is? Anybody?
Pollinated date palm artificially.
Yeah, artificially pollinated. So you should make a note of it. It is artificial pollination outside the naturality. Okay, artificially pollinated date palm. So it is like, just why do we mentioning this? Like it dates back to that time where they have artificially pollinated for the first time date palm. Okay. So next in this hybridization phase, you will see one more example, Thomas Fairchild. So come, tell me what his contribution is.
First artificial hybrid.
Yeah.
Fairchild's mule.
Interspecific hybrid.
Yeah. First artificial hybrid he has developed. So it is a cross between
Sweet William
Sweet
and
William and
Carnation.
Carnation.
Sweet William and Carnation.
Yeah, it is a Sweet William into Carnation. Tell me its scientific names. Sweet William and Carnation.
Dianthus barbatus and Dianthus caryophyllus.
Yeah, it is Dianthus barbatus. It is Dianthus caryophyllus. Okay. So this we famously called it a Fairchild mule. Okay. So another scientist who comes here after that, Easton Shell. What are their contributions?
Ma'am,
Term
Yeah.
heterosis.
They are associated with heterosis. So initially, since these are all like advanced things, heterosis and all comes earlier, what they have done, they have just crossed this maize varieties and developed some hybrids. So based on that, later these concepts and all come. So for this chapter itself, you stick on to this. They have crossed some maize hybrids and they have got hybrids. So they have crossed maize and got the hybrids. Okay. So this you can read, you guys can read. So next is in the hybridization itself, we will see distant hybridization. What is distant hybridization?
It's a cross between two species belonging to same genus or between two genus.
Come again.
Ma'am, cross between two species of the same genus or cross between two genera.
Okay, it is a two different species. You should mention that. Different species of the same genera or different genus. Okay. Tell me the famous examples, primitive examples, which didn't work for this distant hybridization.
Raphanobasica.
Yeah, Raphanobasica. It is a cross between
Radish and cabbage.
radish and cabbage. Yeah. What he has got? What was his intention while developing this material?
Roots of radish intention was roots of radish and leaves of cabbage, but he got reverse.
Yeah. Leaves of cabbage. But he got the reverse. It is a waste. So who is the scientist who has developed this?
Ma'am,
Carpencho.
Carpenter.
In 27.
Yeah. You're correct. Next, we'll come to the somatic hybridizations. Okay. So I'll give you a scenario that these species you are trying to cross are not compatible. There is some incompatibility exists. What you will do? You're attempting this distant hybridization, but you have some incompatibility barriers. So what is the alternative thing you can do?
Mutate.
Huh? Come again. It is a somatic hybridization. What is somatic hybridization?
Crossing between somatoplasm of two different plants.
Yeah, it is a fusion of the protoplast. So we'll take the protoplast and fuse them. Okay, this is the somatic hybridization. So who is the first person associated with this somatic hybridization? Carlson. He's the first person to attempt. Yeah, he's the first person to attempt a somatic hybridization between Nicotiana glauca and Nicotiana longstocki. Okay. So when you hybridize, tell me the products you will receive at the end. So what you will get when you carry out this hybridization in self-pollinated crops? In terms of variety, hybrid, pure line, clone, what do you get?
Pure line.
Pure line.
Okay. What else?
Clone.
Clone you will get in self-pollinated crop.
Variety.
No.
Multi-line.
Come again. I didn't hear you. Okay. Tell me, we will get hybrid or not? This I want to answer. In self-pollinated crops. It is not only in the cross-pollinated crops that we get hybrids, it is also there in the self-pollinated crops. Don't stick on to your cross-pollinated crops when you hear the sound that hybrid. It is also there in the self-pollinated crops, okay? So in cross-pollinated crops, again it is hybrid. What are the other two things when you go for random crossing and all you will get? I have not mentioned here.
Inbred.
Inbred is before hybridization phase. This is after the product after you hybridize something, I'm asking.
Ma'am, after breeding? Or
Just stick on to
like
this hybrid. Okay.
like
You will cross
composites we get from cross-pollinated crops.
Yeah, composites. Composites and
Synthetics.
Synthetics.
synthetics. These are the products when you go for hybridization you will get. Okay. What about this asexually reproducing organisms like
Clones.
asexually reproducing plants? You will clones in that. So these are the products. It is like you just have to know what are the things you will be having. You don't have to stick on to hybrid for cross-pollinated crops only. It is also there in the self-pollinated crops. Know this. Okay. So these are the products, and this is the distant hybridization phase, which is attempted by Raphanobrassica, which was attempted for Raphanobrassica by Carpencho. Okay, let us move on to the next phase. So the fourth phase. What is the fourth phase?
Genetic variation.
Yeah, genetic variations. From where do you get the genetic variations?
Mutation.
Through the mutation. Even through the natural hybridization we get, but it is through the natural hybridization. Okay. So induced is one, which we induce, that mutations. So who is the person? Okay, let us tell what are the rays we will use. X-ray, gamma, alpha, beta, UV rays. These are the rays which we use to induce the mutations. So who is the first person who discovered the mutagenic effect of the X-ray?
Mueller.
It is Miller.
Adler.
Come again. It is Muller. He has given the mutagenic effect of the X-ray. Okay. So from where else we can get this mutation or genetic variations? Not mutation, genetic variations. From where else we can obtain this? Have you heard of somaclonal variations?
Ma'am.
Yeah, from there also, only variations. From there also, we will get to see the genetic variations. Okay. So this is like using the novel genetic variations either by creating or which is already available to the breeding phase. Okay. The next phase comes. Let's see. So Muller has discovered this mutagenic effect. And this first mutation breeding program was started at this you guys should read. Okay. It is started at Sweden. So the next phase is last phase. What is the last phase?
Genetic engineering.
Genetic engineering. What is the basis of this genetic engineering? How does this genetic engineering has come?
Manipulation in the gene.
Manipulation of genetic.
Okay. Manipulation. Manipulation of the genetic material. What is the most basic thing which led us to do this genetic manipulations? What is the most basic thing?
Totipotency.
Yeah, totipotency. What is totipotency?
Ability of a cell to develop in their complete organ.
Your voice is not clear. Come again.
Ma'am, inherent capacity of the cell to develop into a complete plant.
Yeah, it is ability to de-differentiate and again redifferentiate. Okay. So because of this novel property of the cell, plant cell, we could able to achieve this genetic engineering phase. Okay. Without this, we couldn't able to do anything. Even if we make some changes into the cell, how do you make it into an organism? Only because of this totipotency property, we could able to make some changes in the plant genetic materials. Okay. And what else we will have in this? So for what are all the reasons we will do this genetic engineering? Why do we need this genetic engineering?
To create genetic variation.
Yeah, to create genetic variations. Okay, in terms of what are the examples of?
BT cotton.
Yeah, BT cotton. So either for disease resistance, insect resistance, so whatever. If you don't have any variation in your genetic material, you bring that gene which is having a resistance from somewhere, some source, and put it into your plant material and make it into a new resistant plant. This is the basis of the genetic engineering. So in the hybridization phase, in the novel genetic variation phase, that is a fourth phase, I forgot to say something. Novel genetic variations. So one thing we have created through the mutation, somaclonal variation. And one more thing is there, that is a polyploid. Through polyploidy also, we will get to see the variations. So what is polyploidy?
Multiple set of genome.
Yeah, whole genome will be present in the multiple sets. If it is of the same species, what do we say?
Autopolyploid.
Yeah, it is a autopolyploid. If it is from the different species,
Allopolyploid.
allopolyploids. So, any example for this polyploidization?
Wheat.
Yeah, wheat is there. So when we date back, we are looking at the history. When we date back to our history, the first person who has done this is a Clausen and Goodspeed. Clausen and Goodspeed. So you remember this name. This is very important. What is his contribution? Anybody? He has crossed Nicotiana glutinosa with Nicotiana tabacum. Okay, what was the product he has obtained? Nicotiana diglutan. So through the polyploidization also, we can get to see the genetic variation. Okay, and how this process was possible. We know that when we cross between two polyploidy species or whatever, let us take this only, Nicotiana glutinosa with Nicotiana tabacum. So we will get to see the F1. If it is let us take AAV and BBB. It is an hypothetical example. What you will get? Some AB kind of thing. So it will be sterile. To make it fertile, what you will do?
Chemical treatment like colchicine.
Chromosomal doubling.
Yeah, colchicine is very important with this polyploidy breeding. So who discovered this colchicine?
Blacksey and Nebel.
Okay.
Blacksley and Nebel. They have discovered the colchicine. This is also one of the important part. Okay. So this is polyploidy. Clausen and Goodspeed, who has attempted the cross between Nicotiana glutinosa and tabacum to get the Nicotiana digluta, and Blacksley and Nebel, they have discovered this colchicine. And then genetic engineering phase will come. It is because of the totipotency we could able to do this. So let us see the history of plant breeding in India. So it is like a kind of years you have to remember this institutes and all. This I won't be going any brief. You should remember the years because when you aim to come to the IRA, you should know this IRA things. Okay. So in 1905, IRA was established. It was initially named as the Imperial Agricultural Research Institute. It was earlier located at the Hosa. Now it is in Bihar. Like now it is known as Bihar. So due to the earthquake, it is shifted to New Delhi in 1936. And later it is renamed as a Indian Agricultural Research Institute. Same goes with this Imperial Council of the Agricultural Research, which is in 1929. Again, it is renamed in 1946 as a Indian Council of the Agricultural Research. Okay. So in 1921, this whatever the notes we are giving, it is according to BDC. So you and also combined with the Pundansing. Okay, you can stick on to your BDC based on this points wherever we have highlighted. You can read those points only. Okay? 1921. This is Indian Central Cotton Committee, and in 1956, project for intensification of the regional. Research on the cotton oilseed and the millets. 1957, All India Coordinated Maize Improvement Project was started in 1960. First Agricultural University of the Pantnagar. You know this point, First Agricultural University.
Yes, ma'am.
Okay, we'll move forward. So let us come to the objectives of plant breeding. Why do we need this plant breeding? What are our objectives when we start this plant breeding? Come, quickly say.
Crop improvement.
Increased yield.
Higher yield, quality traits,
More yield,
Quality,
quality increase, disease resistance,
disease resistance,
insect,
insect resistance,
agronomic
CUET
traits,
agronomic,
seed shedding, shattering,
agronomic traits. Next,
drought resistance,
drought resistance. Okay, abiotic stresses.
early maturing varieties,
Yeah, early maturing. Early maturing,
non-shattering genotypes,
non-shattering genotypes. Next,
photoinsensitives,
photoinsensitive. Next,
Stress tolerant.
stress are done. Abiotic stress and abiotic stress.
Okay.
Uniform maturing varieties.
Uniform maturity.
A removal of anti-nutritional factors.
Anti-nutritional factors. Or the toxins. Then
Mechanical harvesting.
mechanical harvesting. Okay. So in terms of yield, this is our first and foremost objective we keep, because to feed the increasing population, we need a increased food grain production. Okay. In terms of quality. So why do we need this quality? Nowadays, you can hear this term of very
nutritional
frequently.
security,
Yeah. To empower with nutrition. So to provide the nutritional security, we need not only the increased food, but a quality food. Okay. So this is an insect resistance. We know this from many a times that this is an insect resistance is a continuous thrust. We are empowering the plants to resist the disease and the insect continuously. Okay. In terms of agronomic traits like reduced height, it could be a semi-dwarf, or it could be an increased height also. In terms of where do we need the increased height of a plant?
Fodder crop.
Fodder crops. And also
Border.
jute. Yeah, fodder and jute crops, we need the increased plant height. There we have different kind of objective. In terms of rise in weight, we need a semi-dwarf. So abiotic stresses like salt stress, mineral stress, cold stress. Okay. For all, we will breed the materials. And early maturity. Why do we need this early maturity?
reduce the duration, etc.
We can reduce the crop duration.
Disease escape is
Yeah, disease escape is also possible by early maturing varieties. And we can have a 2 to 3 crops in a year. Okay. So non-shattering. Earlier, we have a very big problem of this shattering. Whenever the grains mature, they would shatter and get lost. So now, due to the development of this non-shattering trait, so we could able to retain the grains. So we could able to achieve the increased yield. This is one of the important reasons. Photoinsensitivity. What is this photoinsensitivity?
not dependent on tail length,
Yeah, not dependent on its day length. Why it is important?
So that it can be grown throughout the year.
Yeah, it can be grown throughout the year, and also we can able to get the early maturity. Okay, we'll come to flowering early. So uniform maturity. Where do we need this uniform maturity?
Pulses.
mechanization,
Mechanization. In which crops?
Pulses.
Yeah, in pulses it will be a problem. like green gram,
Green gram.
black gram. So upon maturity, or they'll continuously maturing. Like we will have to do 3 to 5 pickings in them. So if we get a uniform maturity, if all of them gets mature at a single time, we could able to harvest them at a single time. So anti-nutritional or a toxic substance we have to reduce. So what are the examples for this?
Groundnut.
Groundnut. What was that?
Productions.
Aflatoxins.
Aflatoxin. And what else?
Soya.
Trypsin inhibitors in soybean.
Come again. Trypsin inhibitor. Okay. Trypsin inhibitor. Next. Part of BOA. What is this BOAA? Where do you find?
Lathyrus.
Lathyrus sativus.
Benzoxylin acetic acid.
So it is found in the Lathyrus sativus. What is its common name? Kesari. Okay. This is also we are trying to reduce. And one more. When you heard of mustard, what is the anti-nutritional thing you want
Erucic
to
acid.
Erucic acid. Yeah. So we have to reduce the. This will be one of our objective. So mechanical harvesting. So when we get the synchronous maturity, we can able to do this. Okay. These are all the objectives of the plant breeding. So you can read this. And one more thing, dormancy. So we have one more important thing, that is a dormancy. Why do we need dormancy? To prevent the
unfavorable conditions.
wild bad. So earlier what plants used to sprout on the, seeds used to sprout on the plant itself. So in order to avoid that thing, we need this dormancy factor. Okay. So next is activities. Coming to activities of the plant breeding. So let us give a overall picture of what we do in this plant breeding. Okay. So we will define our objective. Just now we have done this. So if we want some disease resistance. Let us take disease resistance is our objective. So first thing you have to do is assemble the germplasm and or the creation of the genetic variations. So how do you assemble your germplasm, or how do you create your genetic variations? You have two means. Either you can exploit the naturally existing variability, or you can go for the creation of the new variability. You can create for yourself, or you can take from the naturally existing one. So what are the examples? Through the domestication, you will get some crop plants which are having a resistance, or through the germplasm collection, you can get the plants which are having resistant genes, or you can introduce it from the somewhere else place if it is not available in your native place. Okay. Okay, it is not available in the nature. What you will do next thing, you create it for your own. Okay, how do you create? We have seen that we can create it by the hybridization. Either you can go for intervarietal hybridization, that is crossing between the two varieties which belong to the same species. Okay. Or you can go for the distant hybridization. If it is not possible, you can go for the somatic hybridization. The next thing, how you can create the variation is through the mutation, induced mutation, or through the polyploidy, that is autopolyploidy or allopolyploidy. Okay. So you will get the somatic variations where? Through the
Tissue culture.
Yeah. Or you can go for the genetic engineering also, where you will create the genetic variation by bringing the gene from somewhere else. Okay, genome editing. So this is the latest technology we use to create the genetic variation, where we will induce the editing of the genome, precise editing of the genome, okay, to induce the changes. So you have the set of materials. Let us say you have 10 plants, which you find, which are having variation for this disease resistance. So you will go for the selection of them. How do you select? Either through the phenotypic selection, you can see them whether it is resistant or become susceptible based on the symptoms. You can go for this conventional approach of phenotypic selection, or you can go for, which is very famous nowadays, that is a molecular approach, that is a marker-assisted selection. What do you mean by marker-assisted selection?
Using molecular markers, we select the place.
Yeah, it is either you can use the morphological markers or a biochemical markers or a molecular markers. For the indirect selection, you are not selecting the plant directly. It is an indirect selection of your favorable plant based on this marker trait association. Okay. So based on that selection, out of 10, you have selected 5. So you will go to evaluate them. So you will grow them and evaluate which are actually giving the resistance. Okay. Then you have your resistant plants. Out of 5, you will select 2. So finally, you will multiply and make it 100 and distribute it. Okay. This is how you plan your objective and you will achieve your end in this plant breeding. This is the whole activity you will carry out in your breeding. Okay. So what are the achievements? So we have done the breeding and we have achieved something in our past. Achievements of plant breeding. So what is a famous thing that comes to your mind when you say about the achievements?
Green revolution.
Green revolution. What is green revolution? Or with which crop it is associated?
Wheat and rice.
And rice.
Wheat and, oh sorry, wheat and rice. Okay, in wheat, who is a person associated with this green revolution? What is the year of green revolution?
That is
1966.
19?
66.
65, 66, you can see. So, who is a famous person who is associated with green revolution?
Swami.
Norman E. Borlaug.
Norman E. Norman E. Borlaug. So, what was his contribution?
Development of semi-dwarf variety.
Semi-dwarf varieties. What are those? Semi-dwarf wheat.
Sonalika Kalyan.
Wheat varieties. Sonalika
Kalyan Sona.
and Kalyan Sonam. Okay. What was the parent material which has given the semi-dwarf gene for this?
Norin.
Come again?
Norin.
Yeah, Norin. So from where this Norin has come?
Japanese variety.
It is a Japanese variety. Okay. What else?
Semi-dwarf rice.
Semi-dwarf rice. Rice. We are talking about wheat. Okay, let me give you a brief idea about this green revolution. Okay, this is the information you guys know. Let us start. So it is Norman E. Borlaug. Keep this in mind. Okay, so green revolution. What he is called as?
Father of green revolution.
Yeah, father of green revolution. So he is also called as the forgotten benefactor of humanity. He is also called as a forgotten benefactor of the humanity. So Norman Borlaug was born in 1917. So his journey from 1917 to 1965 till Green Revolution. We will see. He has done his BS degree in forestry. This is like an extra kind of thing. You guys can remember few of the important points, okay? So he has done his BS degree in forestry, master's in forest pathology, and his PhD in pathology and genetics. Okay. So he has completed studies in 1942. So after 1942, he joins this Rockefeller Foundation. Okay. As a breeder. He has joined as a wheat breeder. So this was there in the Mexico. By that time, in Mexico, there was already a program which was started in 1943. For Green Revolution, they have already started a program that is known as a Mexican Agricultural Program. So in this program, they have aimed to increase the food production to feed their population. Okay, they have already started this Green Revolution kind of thing. So to their Norman E. Borlaug has joined. So he has become a head of that program. So he led this program, which finally resulted in our Green Revolution. Okay. So coming to materials, let us take the materials. This is okay.
Excuse me, ma'am.
Huh?
Ma'am, why he's known as the forgotten benefactor of humanity?
Because this thing I will look and tell you. Okay.
Okay, ma'am.
So he has added this Mexican agricultural program. So for the development of this varieties, he has collected the germplasm worldwide. Okay. He has collected the germplasms worldwide. In that collections, he has got this Norin 10 and also a cross between Norin 10 and Brewer. There, F2 segregating material he has got. These are the germplasm collections which has obtained from worldwide. He has identified two materials, that is Norin 10 and a F2 material which is a cross between a Norin 10 and the Brewer. Okay. So this Norin 10, it has come from the United States Department of Agriculture. So it was sent by the Samuel C. Salmon. So this particular material, he is a very important person. You should remember this. Samuel C. Salmon. He has provided this Norin 10 material for the Norman E. Borlaug. Okay. This cross material was provided by Ogle or Ville. Ogle has provided this cross material. So using these two materials only, further he has developed this Lerma Rojo and Sonara 64. So these are the two varieties which has led to the increased food production in Mexico. Okay. Meanwhile, in between also in 1965, CIMMYT was also established in Mexico. Keep this point in mind. Okay. From Mexico only we will get the materials to India. Okay. So you guys have got some idea. He has joined this Rockefeller Foundation as a breeder in Mexico. there he has started breeding for wheat. So for that, he has collected a germplasm from worldwide. In that germplasm, there are two important germplasm, that is a Norin-10, which is a source for our dwarfing gene, and also a F2 cross material of the Norin-10 and the brewer. So this Norin-10 is given by the Samuel C. Salmon from United States Department of Agriculture. And this crossed material was given by Orville and Vogel, V-O-G-E-L. So these two are the important scientists you should remember, okay? So from that only he has developed this Lerma-Rojo and Sonara 64, which has led to a green revolution. So later these materials are introduced into the India, Pakistan, and one more place they have been introduced, Turkey. Okay. Turkey in 1966. So from there only our journey of green revolution has started in India. Okay. So from these materials we have developed Kalyan Sona, Sonalika. Along with that, two more are developed. What are they? Choti Lerma, and Safed Lerma. Okay, so the same kind of pattern is also repeated for the rice. Like they will import some materials for the dwarfing gene, which is a source of the dwarfing gene, and they'll cross with the native materials which are available. He has crossed these two materials with the materials which are already available in the Mexico, okay, to develop this Sonara 64 and Lerma-Rojo. Remember this. He has crossed with the Mexico materials. Like that only for rice also, they have crossed with some native materials and come up with the semi-dwarf rice varieties. So the same kind of pattern has also appeared for the rice. This is how the green revolution has started. This is how the materials for the green revolution has come. Okay. So semi-dwarf wheat was developed by Norman E. Bowler, and the dwarfing gene source is from this. Sorry. So this is a variety. You should not confuse with this. Okay, Norin-10 is a variety. So what are the genes which are available in this Norin-10? It is RHD1 and RHD2. And also along with that, we have also found this RHD3 and RHD8, but they are associated with the lodging resistance, not with increasing the yield. They were giving the lodging resistance to the plant. They're also associated with increased yield also. Okay. What is their mode of action? How this RHD gene will reduce the plant height? By encoding the Della domain proteins. Okay, Della domain proteins is a kind of inhibitory proteins. So for any plant, if it produces a gibberellic acid to become responsive, it doesn't directly act. It will go and trigger some of the genes which will respond to this gibberellic acid. So what this Della domain will do, it will target these genes. It will prevent the transcription of the genes which actually respond to this gibberellic acid, so that there won't be any, even if gibberellic acid is present, there won't be any response to this gibberellic acid and the height of the plant doesn't increase. This is how this RHD gene acts, okay? You will also get SD1 in case of rice for the reduced height. So there, this SD1, it directly stops the synthesis of GA itself. Okay, you remember this mode of action of these two genes. For reduced height in rice, SD1 is there. It will reduces the GA biosynthesis itself. Whereas in case of wheat, RHD1, it will encode a Della domain inhibitory protein, which will prevent the action of the transcription of the genes which respond to this gibberellic acid. Okay. Let me ask you one thing. Who has given this term Green Revolution?
Swaminathan.
Term Green Revolution. No. Term. Have you heard of William? Come, I'm giving you hint. Tell me somebody. William?
William, yes, God.
Yeah, yes. God. William is God. He has given the term Green Revolution. Okay. So along with this reduced height, RHT1 and RHT2, we have also photoperiod insensitive genes in this wheat. What are those?
PPD.
Yeah, it is
PPD1.
PPD1, PPD2. So what is the optimum combination of these two genes to get a better yield?
RST1 plus PPD1.
PPD1, or you can have a PPD2, but never the both, like RST1, PPD1 and PPD2, because they won't function properly. Any of the combination, they'll work for optimum yield. So if both are there, that will lead to some kind of malfunctioning. Okay. So I have a question. From where this Kalyan Sona and Sonalika has come? From which material we have developed this Kalyan Sona and Sonalika? Just now I have told.
Lerma-Rojan Sonara 64.
Sonara 64.
Lerma-Rojan and Sonara 64. From where they have come?
Mexico.
Mexico.
Yeah, you should remember this point. CIMMYT, Mexico. It is a selection from the material. CIMMYT, Mexico. These two are developed. Okay. So next, let us move on to the Green Revolution in rice. So you don't have much in this rice to remember. I don't say any history for this. It is the same kind of pattern that some material they introduce into the native places or wherever the work is going on, and they'll cross with the material which is already available with them, and they have come up with our Green Revolution varieties. What are the Green Revolution varieties of rice?
IRA.
Matching native one.
Matching native one.
TN1.
Yeah, good. So I'll say one of the person name, Henry M. Mitchell. So you should remember this thing. It is very important. Okay. So he has developed this world first semi-dwarf IR8, semi-dwarf right IR8. Okay. And also we have GH Kush. What he is famous for? IR36. Okay. When you come to rice in terms of Green Revolution, you should remember these two names. And also Swaminathan is also there, MS Swaminathan. We will have a separate kind of interactive session for this, not now. So in case of rice, what is the source of dwarfing gene?
Digivogen.
Yeah, dwarfing gene is from the, I'll write on this part. Digivogen. So what is the gene?
ST1.
Yeah, ST1. What is its mode of action?
Prevents gibberellin cells.
gibberellic. It will block the gibberellic acid biosynthesis. Okay. So what else we have in this? We have IR8 and TN1. So for them, what is the source? It is DGO Ugen for the SD1. Okay. So IR8 is a cross between, anybody know? How do we obtain this IR8? IR8 is a cross between DGO Ugen and Sai Yuan Chung. CHUNG. It is UNG. Sai Yuan Chung. So it is a cross between this DGO Ugen and Sai Yuan Chung. We have obtained as IR8. And TN is a cross between. Oh, sorry. This is actually TN1 cross. IR8, I will tell you. IR8. This is for TN1. Sorry. IR8 is a DGO Ugen with beta. Okay. You should remember this process also. IR8 is developed by DGO Ugen into beta, and DGO Ugen into Sai Yuan Chung is a TN1. So this was developed in place. Anybody know?
Taiwan.
Yeah, it is in Taiwan. This is in
IRRI Philippines.
Yeah, Philippines. It is in Philippines. So this is the information you should be knowing in this rice breed integration. Okay. I have one more question. From this, we have finally, after entering into India, we have developed Jaya and Ratna from these materials. Okay, these are the selections from these two. This part you should remember. Anybody know what is the cross of Jaya?
TN1 and type 141.
Yeah, TN1 with type 141. Very good. And also two more varieties are there. IR 24 and IR 36. So these are all the varieties which are associated with the green revolution. IR 8, IR 24, IR 36. Okay. I told you IR 36 is by who?
GH Kush.
Yeah, GH Kush. Okay. So this one more question is there. Who is the father of hybrid rice?
Yuan Langping.
Yeah. She is not there. Yuan Long. Okay, he is the father of the hybrid rice. So this is about the first achievement when we look back to our history, that is a green revolution. Okay, this you can read. So the next achievement, what is the next achievement we have in our history? Mobilization of
sugarcane.
Yeah, mobilization of sugarcane. What is this mobilization of sugarcane? So,
Increase the yield of North Indian canes.
yeah. So they had initially two kind of canes. That is a tropical and the Indian cane. So this Indian cane was having a lower yield and lower sugar yield also. But it is hardy in nature. Like it is having some kind of disease resistance, pest resistance, or it can able to tolerate the abiotic stresses. It was hardy. What was its scientific name?
Saccharum barbari.
Saccharum barbari. Yeah. So coming to tropical cane, what is the scientific name?
Saccharum officinarum.
Okay, see, narrow. So what is its nature? It is having a higher yield. It is having a higher sugar yield also. It performs overall performance as well. But what they have observed when they have taken this tropical canes into this North India, but they did not perform well. So for that, what they have done, they have crossed this tropical cane with the Indian cane to come up with the sugarcane varieties which can perform well in the North India, associated with the hardiness of the North Indian canes and also higher yield and higher sugar yield of the tropical canes. This is how they have this crossing program and making them available for the North Indian conditions also. They have called it as a nobilization of the canes. Okay, nobilization of canes. So who is the person who has done this?
T.S. Venkataraman and C.A. Barber.
T.S. Venkataraman and
C.A. Barber.
C.A. Barber. Good. So he is a person who is associated with this. So one more sugarcane variety is there, Saccharum
Spontaneous.
sinensis. Okay, Saccharum spontaneum. Tell me, what are they? What is this and what is this?
Chinese cane.
Disease resistant.
Disease resistance. Okay, where it is grown?
China.
Yeah, it is a Chinese variety. What is this spontaneum? It is a wild sugarcane. It is okay. It is having a genes for the disease resistance. Okay, this is about the nobilization of the canes. You can read it. So next, the third achievement is with the development of the hybrid millets. Or hybrid development. So let us take maize. When was the first maize hybrid was developed? It is in 1961. What are the four famous hybrids which were released in that time? Anybody remember?
Ganga.
Ganga.
Ganga 1.
Ganga.
101. Then
Deccan.
Yeah. One more.
Deccan 103.
Ranji. Okay, these are the four hybrids which are released during 1961. And coming to Bajra. When was its hybrid released?
1965.
1965. No, it is in 1964. So HB1 and also for the sorghum also released in the same year, 1964. This is sorghum hybrid 1. Okay. So there are many other hybrids also, and also many other varieties like composites, synthetics are also there for maize that you can read and remember. I have mentioned here. This is about the hybrid programs. Okay. So the next is the hybrid cotton. The fourth achievement is the development of the hybrid cotton. First, tell me what are the four kinds of cottons which are there with their scientific name. I will give you a clue. We have a tetraploid maize. Sorry, tetraploid cotton. And a diploid cotton. What are they?
Tetraploid, Barbadans, and Hirsutum.
Gossypium barbatus. Gossypium.
Hirsutum.
Excuse me.
Hirsutum.
Oh, sorry for the disturbance. Come again. Gossypium barbadense and
Gossypium hirsutum.
hirsutum. Diploid?
Gossypium arboreum and Gossypium herbaceum.
Arboreum and
Herbaceum.
herbaceum. What is its ploidy? 2 n is equal to
26. 52. 52.
Good. So, what is the first cotton hybrid which was developed?
H4.
H4. So, it was developed in 1970. It is a cross between
G67 and American Netherlands.
Gujarat 67 and American Nectarines. Okay. So, this is it is intra or inter?
Intra.
Yeah, it is a intra hirsutum or an intra-specific cross, you can see. What is inter-specific cross?
Varalakshmi.
Yeah, it is Varalakshmi. It was developed by cross between. It is a Gossypium hirsutum into barbadense cross. Okay, developed by.
US Dharwad.
Okay, US
Dr.
Dharwad.
Katarki.
Yeah, DH Katarki. Okay, it was developed by Katarki. So, and also you have some other examples. You can read it on your own. And the first Desi cotton is Gossypium arboreum. These are the two hybrids of the Desi cotton. You can remember these examples, okay? So next, coming to molecular breeding, there is nothing much in this. So through the marker-assisted selection, we will go for the indirect selection of the trait or the variety which is showing association with that trait, okay? Through that, we have developed this improved Pusa Basmati 1 and improved Samva Masuri for the bacterial blight resistance, and Swarna Sub 1 for the submergence tolerance, and Vivek QPM 9 is for the improved protein quality. This is one of the achievement. Okay, for transgenic varieties, we have this BT cotton for insect resistance cotton, and Bikaner Narma, it is an indigenous BT cotton, and NHS4 is a hybrid BT cotton. Okay. So let us discuss this. What are we have done? We are deliberately doing this breeding program from past many years. So unknowingly, we are doing some of the mistakes, or we are deliberately having some consequences on these genetic materials. What are those? So what are the consequences of this plant breeding? So the first thing is the genetic erosion. So what do you mean by genetic erosion?
Loss of variations.
Yeah, loss of genetic variability due to, it can be due to loss of genetic variability due to increased use of the uniform varieties. Like you will replace your land races which are having a valuable genes for disease resistance or whatever, which is a heterogeneous material, which is heterogeneous. You will replace them with the improved varieties like your pure line or any of the hybrid, which are highly homogeneous. Okay. So when you replace this heterogeneous material with a highly homogeneous material, unknowingly you are eliminating the variability which was present in your material, earlier material. That is leads to the genetic erosion. So let me have one question. And also use of this similar varieties as a parent. Use of similar varieties as a parent. When you use the same parent, same variety, let us take variety 1 as a parent in all the crosses. Like for P2, for P3, for P4, you are invariably making them as a uniform. One or the other way, they have some uniformity related to them. So that is how you will lead to the genetic erosion. Okay. So what is a genetic base? What do you mean by genetic base? And genetic vulnerability. These two terms. Can anybody say? Genetic base and genetic vulnerability?
Ma'am, genetic base, mainly the genetic variability present
In your cultivated variety. Okay. So what is the genetic variability? Variability present in your cultivated crop material. That is your genetic base. So what is the genetic vulnerability?
Ma'am, it is the susceptibility to disease or insect attack.
Yeah, susceptibility to the disease or insect attack, mainly due to the uniformity. Mainly due to the uniformity of these varieties, they'll become susceptible to some of the disease and pest. Then we will see this genetic vulnerability. Because you have used the same resistant parent to cross with many of the varieties, hundreds of the varieties you have developed. So by some chance, the pathogen was able to mutate and counteract this resistant gene. Then it will become susceptible. Then all your 100 varieties which are used for developing these varieties will become susceptible at a single time. Then you will see a loss in your genetic variability and loss of your crop also. That leads to a genetic vulnerability. Okay. So narrowing of this genetic base. So what do you mean by narrowing of genetic base? Genetic erosion, you can read this. So tell me, what is narrowing of genetic base? Second consequence is unknowingly we are narrowing our genetic base. Genetic base, we know what is the variability which is present in our cultivated material. That is genetic base. So we are narrowing it. What do you mean by that? I'll give you a quick example. We have a DGO-Ugen. It is a source of dwarfing gene for TN1 and IR8. So these two have served for many of the varieties which come after this green revolution as a SD1 source. Okay, so I can say that all these materials are related to IR8 and all these are related to TN1. But knowingly or unknowingly, these two are itself are related to DGO-Ugen. That is, they are not much diverse. They are also related. These are also related. If any one becomes susceptible, then all its lineage will become susceptible, right? That is how earlier it was like this, but we are doing it as a narrowing it this genetic base. Okay, which eventually leads to a genetic vulnerability. So what is a famous example for this? Genetic vulnerability caused by narrowing of the genetic base. You have heard about CMS T cytoplasm.
Texas maize.
Yes,
Yeah, it has become
Yes.
susceptible to
Southern blight.
Southern corn blight.
yeah, southern corn
Helminthosporium made.
Helminthosporium malis. Okay, so that time, whatever the hybrids which were developed, they are developed by using this CMST cytoplasm. So once this cytoplasm has become susceptible to this Helminthosporium malis, all the hybrids which were containing this CMST cytoplasm have also become susceptible, which has led to the epidemic condition. Okay, this is how the narrowing of genetic base will take place. One more example is there, I'll show you. So Bajra downy mildew, it is due to the cytoplasm TIF23A, but it is not due to the cytoplasm, it is due to the nuclear genes. So while crossing, you will do repeated back crossing, right? So few of the nuclear genes will remain, even if you do many of the crosses, at least few of the genes will remain. So those genes have encoded the susceptibility for this Bajra downy mildew or ergot. Okay. So,
Ma'am,
and
ma'am, could you please repeat it?
Actually, it is like you have to think it hypothetically. It is not directly written. Like we are saying, it is exactly by the CMST cytoplasm. Okay, when we use it as a female parent, it will give only the cytoplasm, but they have mentioned it as a nuclear gene. It is due to the susceptibility of this cytoplasm associated with nuclear gene. That means there are some nuclear genes which are transferred from this female parent, which has given our cytoplasm TIF23A, right? You cross it with your another variety which you want to make it as a hybrid. You will get this hybrid. Okay. Not because of this cytoplasm it has become susceptible, because of the some of the nuclear genes which has got from this female parent, you have got this susceptibility. Okay, you have got it.
Yes, ma'am.
Okay, so next is a increased, another susceptibility, the increased vulnerability to minor diseases. So we know that over a period of time, breeding the crops for major diseases, we have neglected this minor diseases. So in the course of time, they have also evolved and they have also gained much of importance that now they are becoming the threat. So that is our vulnerability to the minor diseases. So that, for example, you can see Uganda 99, which is a, you just have to remember these points, okay? It is a super risk of the stem rust. So one of the examples for this minor disease which has become major is a gray mold of chickpea, which is caused by Botrytis cinerea, kernel bunting weed, which is caused by Tilatia indica. Okay. So and another problem which we face in the future is the yield plateau. That is, you will get exhausted with whatever the variability you can what you can use for creating the increased yield. That will become exhausted at some point of time. Then you will achieve a plateau in your yield. There won't be any increase unless you come up with new methods to create the variability. Okay, that is a exhaustion of all the available variability for increased yield. Then you will reach a plateau in your yield. Okay, after that, if you need any increase in that, you will come up with you have to come up with new methods to create the variations. Okay.
Ma'am?
Yeah.
And then how to achieve increase in yield without yield plateau?
This is like we are seeing in terms of history. We have already achieved this. We are continuously increasing. We have come up with the genome editing technologies also. So we can break this. It is in terms of history we are seeing. Now in our present, we have already broken this. We could able to achieve. Okay.
Okay, ma'am.
So I have some other miscellaneous examples for you. IR8 and TN1. Sona and Sonalika, famously known as the forerunners of the Green Revolution because they are the first derived varieties. Okay. So one more example is soft rot of chickpea, which is caused by Rhizoctonia betaticola. That is also becoming a major threat nowadays. So finally, I have a list of Indian plant breeders. This you have to memorize. Okay, what is the crop they are associated with? If they have given any of the important varieties, you have to remember these things. Okay. So this is for your memorization. So T.S. Venkataraman, you know this. He is associated with mobilization of cane. B.P. Pal, he is associated with the wheat. Okay, he has developed this NP series of wheat. Okay. M.S. Swaminathan, I will have a discussion with you guys about M.S. Swaminathan later. So he has developed, he was also associated with the Green Revolution. He has developed this eye yielding wheat and rice varieties, and he is known as the father of green revolution in India. Okay, he is in India. You have to remember this. So Pushkarnath, he is associated with potato breeding. He has developed what he has given some of the famous technique. What is it, Pushkarnath?
Seed plot.
Yeah, seed plot technique. Why it is used?
Virus elimination.
Yeah, to prevent the virus infection. Okay. So we will cultivate for seed potato. We will do the cultivation in such a period of the year where the aphid infestation is very low to prevent the transfer of this viruses. Okay. So S. Ramanujan is associated with the true potato seed. And NGP Rao, he is a sorghum breeder. K. Ramaya, GH Kush also you have to remember. And one more person, tell me. With rice. I have told you in Green Revolution. Anybody remember?
Henry Beechwell.
Yeah, Henry Beechwell. You have to remember these names. So one more is K. Ramaya. He is associated with India. Okay, Ramdan Singh, it is very important. He is a wheat breeder. For our SRF exam, they have asked this variety. So you have to remember C591. Okay, D.S. Athwal is associated with the pearl millet breeding. Bosison with the maize breeding. Dermpal Singh, oilseed breeding. C.T. Patel, you know already, that is the father of hybrid cotton. And we, Santhan, upland and the Egyptian cotton. Okay. So this is about the brief introduction about your introduction to plant breeding. So we have seen on a surface level what happened in our past. So in the moving classes, you will see many of the techniques, many of the scientists and concept-based things. So in this chapter, you have to memorize many of the things and the varieties names and scientist names. You do that for now. We'll have the next class. Thank you. If you have any doubt, you can ask me here, or you can write in the chat box, or you can write in the WhatsApp also. If anybody have any doubt,
No, ma'am.
okay. Thank you. Thank you for listening. Happy New Year.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am. Happy New Year.
Happy
Thank
New Year,
you.
ma'am.
Thank you.
Enjoy your last day of 2023. Happy New Year. Okay, we can end the class then.
Thank you, ma'am.
Thank you.